10 years ago, when Emily was not responding to any type of chemotherapies in her battle with cancer, the doctors and scientists decided to take a new approach in which they collected her blood, engineered her immune cells to become smarter and stronger, and then put them back into her body. And that was when the miracle happened. Last month was the 10th anniversary that Emily has been cancer-free. For the field of cancer immunotherapy, to create more of such wonderful stories, there are several challenges to be overcome. This includes reducing the cost, complexity, and time associated with cell engineering protocols, while also improving the efficiency, safety, and precision. I am Mohammad, co-founder of Cellecho Biotech. At Cellecho, we have invented a microchip that uses a combination of acoustic energy and electric field to create hundreds of thousands of tiny little microvortices, similar to a washing machine, that can precisely manipulate cells and deliver genetic coding molecules into them. Our Cellecho technology not only provides high cell engineering efficiency and precision associated with viral-based approach, but it also is a fast, simple, and less expensive method. The Cellecho technology is covered by 3 major patents, which all of them have been recently being issued. Overall, the cell and gene therapy market size is rapidly growing, and we estimate that by 2025, Cellecho can target up to $4 billion of the market for both cell therapy and genome engineering. Our business plan is to partner with pharmaceutical companies as well as academic leaders in the field. Several pharmaceutical companies have already shown their interest in using our technology to provide solutions to their cell manufacturing bottlenecks. In addition, we are currently collaborating with academic leaders to bring the next generation of cell therapies to the market. To achieve this, we are currently seeking to raise $2 million to help us cover the costs associated with business development, operation, and technology development. Here is our team, led by Dr. Abraham Lee, who is a Chancellor Professor in Department of Biomedical Engineering at UC Irvine. He has many years of experience both in industry and academia. I myself got my PhD in biomedical engineering at UC Irvine, and I am currently an NIH-funded postdoc researcher at UCI Institute for Clinical and Translational Science. Thank you so much for your time, and I will be more than happy to answer any question.
Thank you for that. Could you just back up and show us sort of the stage of development you are and what's next from a milestone standpoint?
Sure. So we are currently have already our MVP that we can test our device with immune cells that we have. And right now we are collaborating with both academic leaders and pharmaceutical companies to engineer the cells that they are targeting with.
Sorry, Mohammad, I missed what the device is doing. Can you describe what the device is actually doing?
Sure. So basically, to engineer cells, we have to send the materials that are able to engineer them inside the cells. And overall, the cells don't like that because they have a membrane that is like a castle. They don't want foreign materials to go into them. So we use a combination of acoustic energy and electric field to basically open up the cell membrane and deliver the cargoes into them. So our system is like a washing machine. So you basically open the cells and rapidly mix any materials like CRISPR, DNA, mRNA into the cells.
And so this is not like a standard electroporation. This is for mammalian cell primarily, or?
Yeah, electroporation is another competitor in the market, but our method is gentler and also it offers precision for cell engineering. Yes, but it's being used for both mammalian cells, plant cells, and any other biological components. Yes.
What's your relationship with the university and the research and your company?
So right now we have the patent and we are negotiating with the school to license the patent. And I myself, I'm a postdoc at UC Irvine, but I'm going to transition to the full-time for working for the company.
Can you talk a little more about the precision that you were just saying with electroporation and other methods? Is the brute force way, right? So how is this more precise?
So basically right now, for example, I give an example for cancer immunotherapy. When current method, when they engineer cells to make them smarter, Some cells become super smart, some cells lag behind, they don't become that much smart. And this is because the amount of cargoes that we send into cells varies. Some cells receive 10 molecules, the other cells receive 1,000 molecules. And the unique thing about our technology is that we can precisely mix the cells with the cargo that we want. So the population of the cells that we have, all of them on average receive the same amount of cargoes.
So just maybe quickly, could you contrast, let's say, this versus Squeeze Biotech, where the approach was initially the device, but now it's more therapeutic. So what is the plan you have? Is it more just to sell the device to pharma companies or to actually go that route?
A very good question. Yes, I mean, to be honest, we are exploring different business plans. When I started my PhD and we came up with this idea, our first business plan was to make an instrument and sell it to, for example, research lab or the companies. But right now, as you mentioned, similar to a Squeezy, our business plan is to kind of form partnership with pharmaceutical company. A Squeezy at this point, they are mostly collaborating, for example, with Roche, but we have other pharmaceutical companies that they have shown interest in using our technology. So our technology, our kind of business plan is to be part of their cell manufacturing protocol.
Okay, sorry, one more question. So in terms of data set that you need to demonstrate that your system, I guess because it's more like a single cell, you get a consistent cell-to-cell transfer of, let's say, RNA or DNA. You guys already have demonstrated that there is more consistent transfer of materials to the cells versus existing competitors on the market. Is that correct?
Yes, we have recent publications that we demonstrate that we can precisely control the amount of molecules that we can deliver. Methods such as electroporation or even a Squeezy, they are based on kind of a stochastic delivery. So they are based on the diffusion or random delivery into cells. So our method, we use active mixing to deliver. So we have shown the results in our recent publications.
So if you can go to the slide where you said where the money is going to be spent in different
sure.
areas. I don't know how much time we have. We have time. So what's the difference between operations and technology here?
So for operation, right now, we are, for example, renting their research offices,
Okay.
and also, for example, buying the equipment. For technology, I think the most part of the money is going to go for developing better MVPs and also mass manufacturing as well.
Can you give me a sense of the scale of production? So you talk about at the single cell level, how many cells and what amount of time? Like what's the scale and how can you move up for manufacturing?
A very good question. So the scale right now for cancer immunotherapy, it should be around, for example, 10 billion cells. So we have to engineer in the range of billion cells. Right now, our system, the throughput is, we can engineer 10 million cells per minute. So we are right now scaling up the cells. So in a matter of 1 hour or so, we can reach that range of throughput. But it's a very important factor in engineering cells, the throughput.
Nice work.
Actually, there is one question over here.
It'll help the folks on.
Thank you. Have you approached any prospective partners about the technology? And if so, what type of partners are you, with what type of partners are you getting the most traction?
Yeah, thank you. Thanks for the question. Yeah, to be honest, my background is in mechanical engineering, and we are actively looking for pharmaceutical companies who are very smart in biology and they can target the bottlenecks in the field. So right now, we are actively contacting research labs that they are expert in biology and pharmaceutical companies that their main target is to develop therapeutics that are based on cell engineering. So we feel like partnering with them can help us further develop our technology to address their challenges in the field.
Can I ask one? Yeah. So Mohammad, why not go raise $25 million? Because you're going to need to have a GMP facility to make these devices, right? So if you're going to partner with many different companies, I mean, I don't know if 2 million will get you there, but in some ways, you actually might do better by asking for a lot more money and actually have a long-term plan. So like, what was the logic in thinking about 2 million versus 25 million?
It's a very good question. I mean, we are still in the process of learning, and I mean, it's a very good advice that I can take and discuss with our team. To be honest, this $2 million is basically our estimate based on the funding that we apply for NIH and NSF that kind of gives us a little bit confidence and to move the technology further. But it's a very good advice. I mean, we can further incorporate that. Thank you so much.